All right. Good afternoon, everybody. Good afternoon. Good to see you. Good to see you. All right. Okay, well, welcome to the first real Monday of the semester. Yeah, okay, we're here. We got an exam coming up on Friday, so a couple of things about that. If you didn't have me last semester, I try to make the quizzes somewhat, in the similar format to the exam. So the questions are like that. So there's a group of a lot of multiple choice questions, as well as a group of those kind of questions I call like hybrid. That's usually like a paragraph with a bunch of blanks in it, and then you have a bunch of options, like up to 20 or plus options to put into those blanks. Okay, so that kind of takes the place of like a short answer question. So you're not writing it out, but you really do need to know it at the level of being able to write it out to be able to be successful on those questions. Okay, so and just one thing I would definitely recommend on how to study for that type of material, because this course is really kind of two big types of material. There's the stuff that's just the pure memorization, kind of terms, structures, functions, all those things that were really good for flashcards, right? So people that make flashcards, that's a great thing to do for that kind of stuff. But then you have like the process-based stuff. For example, like the eyes and that whole process that the eyes go through when an object is approaching you or moving further away, right? There's multiple things that happen. Which cranial nerves work and which muscles contracting, why is it doing that? Those kind of like conceptual as well as like step-by-step processes. Can't really use flashcards for that, right? So what I recommend with that is to kind of study it in the notes, read it over and over, put those notes aside, and then have a blank piece of paper in front of you. See if you can just write it out off the top of your head. And to do it in your own words too, a little bit, as well as including some of the correct terms, but to do it in your own words in such a way that, because when you get to one of those hybrid questions, it's not the exact same wording as the notes. And so if you just purely memorize the notes and then you read one of those hybrid questions, you can be kind of lost, right? Some of you had that experience last semester. So I kind of recommend putting it in your own words, kind of making sure you understand it. And yeah, and then you can go back after you write it out, go back to your notes, see how you did, what'd you leave out, study some more, try again, right? It's always better to study. So study, stop, quiz yourself. Study, stop, quiz yourself. That's what flashcards automatically do, right? But when it comes to those longer process-based things, you got to kind of come up with a different way to do that. So that's just my recommendation. But yeah, definitely want to put a lot of time in to studying for the exam this week. What we talk about today will be the last material that's on exam 1, right? And then we will move on to some new material on Wednesday, but that'll be the first bit of information that's on exam 2. Okay, we will use HonorLock. And so if you haven't had to use HonorLock yet this semester, and first of all, everybody's got to take the HonorLock practice quiz, but it's just a way to make sure your computer is ready for it, right? And so sometimes, even if you did it last semester, and there was some update to your browser or something, and then you go to use it and it doesn't work, right? You don't want to be messing around with that on Friday in class instead of taking the exam. So that's why the HonorLock practice quiz is there. Okay, so just hop on there sometime in the next couple of days and do that. If your computer's not working when you're trying it, then use the little chat function right there and contact their tech support. They're super responsive. They'll get on, they'll help you figure it out and get your computer set up. That's the whole purpose of that. Okay, so make sure you do that. Okay, lab this week. We'll have a quiz kind of over just the understanding of the activities that you did last time. Okay, so you can just kind of look back on last week's lab, make sure you understood what you did. If you did, then you should be fine. It's not super, not a super tough quiz, but it's just kind of a recap of that, which also helps you get ready for the exam this week, because most of that stuff's on the exam, right? The reflexes and sensory receptors. Just to be clear though, I don't pull stuff from the lab that's not in the notes and put it on the exam, but there was a lot of overlap anyways, right? So, okay, yeah. What else? Any other questions about what's going on this week? Okay. All right. Okay, well, so today we're going to go over to the last special sense that we'll talk about, which is in the ear. Okay. And we kind of deal with two different special senses there, which is obviously hearing, but then also equilibrium, balance, proprioception, those types of things. So today will be mostly just the anatomy of all the different parts of the ear, and then we'll get into the more of the physiology on Wednesday, and that'll be the stuff that's on exam 2. Okay, so let's dive into some of these structures here. So we'll start out on the most external part of the ear, which is called the external ear. And the part that we can see is called the auricle or the pinna. Okay, so it definitely has some functions. It definitely works as a sound collector, right? It's shaped in that way so it can actually catch sound waves. Okay, and it is full of quite a few sensory receptors itself. And so by the vibrations hitting different parts of your ear, it does actually give you an idea of where the sound is coming from, right? Which is kind of interesting. Certain animals like dogs can detect that and then move their ear towards that, right? We can't do that unless somebody in class has a special talent. Anybody move their ears like that? No. Okay, some people can kind of wiggle them around a little bit, but yeah, we can't do that. But it is pretty funny or kind of cute to watch dogs do that, right? When they're facing away from you, but they're kind of trying to still keep an ear on what you're doing, right? but there's definitely a sensory receptor part of that where they're feeling the sensation of the sound waves from coming from behind them, so then they can turn their ear around and catch that wave, catch that sound, catch that wave, yeah, catch the sound waves. But, yeah. And so then going in from there, we have the auditory canal, which is also known as the external auditory meatus. Okay, this is just the passageway, obviously air-filled. We have special ceruminous glands in there, which secrete earwax, right? And earwax has some important functions for us, mostly repelling things from insects from coming inside of there, and then trapping debris before it gets too deep inside of our ear. Okay. All right, so that's the external ear. And then we'll move into the next canal of the ear called the middle ear. This is known as the tympanic cavity. Okay, this is also an air-filled cavity. All right, so we pretty much have 3 cavities within the ear: the external, the middle, and the internal. The external and the middle are both air-filled, and then the internal is fluid-filled. Okay, so it's lined by mucous membranes all throughout. Okay, and the boundaries of it are, well, it's between the eardrum, also known as the tympanic membrane, and then the oval window, which is on the internal ear side. Okay, so both little membranes that are similar to each other kind of act like little speakers, and we'll talk more about how that works in a sec. But that tympanic membrane is this flattened cone shape pointing medially. So it's not completely flat. It actually has a little point to it in the center that goes in medially towards the center of our body. And then it's going to connect to one of the little auditory ossicles that we'll see in a second. Okay, so it's actually skin on the outside of it, but then on the inside is a mucous membrane. Okay, and so this acts to transmit sound from the external ear through or into the middle ear. Okay, and if you've ever taken the cover off of a speaker or just seen a speaker without a cover on it, then you know it kind of has this flat membrane-like structure. And if you've watched it move when sound pumps through it, and you see that membrane kind of bouncing back and forth, that's exactly what your eardrum does when sound waves hit it. Right? It just moves back and forth, and it's that vibration, the frequency of the vibration, that's going to dictate what that sound sounds like to us. And we'll get into that more next time. Okay, so then inside the middle ear, once we get past the tympanic membrane, we've got these little auditory ossicles. These are the smallest bones in the body. You can see how small they are in comparison to somebody's finger, right? They're just these tiny little bones, but they are pretty beautifully shaped, right? They all have these really unique little shapes to them, and they all connect to each other. They actually all have little synovial joints between them, right? So they have synovial capsules with fluid inside, just like your knee does, just like your elbow does, right? There's synovial, not synovial, but there's hyaline cartilage on the inside of those, just like any other joint would be, right? But they're just tiny, tiny little guys. And they all have kind of things that they look like. Oops. They have little structures that they look like. And so the malleus is the hammer, the incus is the anvil, and the stapes is the stirrup. Okay. And you can kind of see how they fit into each other. But their big role is to transmit that sound from the eardrum or from the tympanic membrane all the way over to the internal ear, which connects there via what's called the oval window. Okay, and one thing that's kind of interesting about these is we actually have muscles that attach to these bones too. And what can happen is there's these tiny little muscles that can tighten up the joints between those auditory ossicles that can limit how much they move. Right, so when we have a very loud sound that hits our external ear, we have reflexive actions that go quickly to our spinal cord and then back to the motor neurons that contract those muscles, and it tightens those muscles up so it doesn't transmit as much of that sound into our inner ear and potentially damage our inner ear. Okay, questions about any of that so far? Go ahead, Trevor. Yeah.
So like as we age, joints kind of just slowly
Mm.
degrade. Is that why like when you're older you're more sensitive to really loud sound?
That's a great question. I've never seen anything related to these bones or these joints degenerating. Doesn't mean that doesn't happen. Typically what happens with hearing is more of a loss of the function of the internal ear. But definitely when we have an ear infection, we'll talk about in a second, when we have anything that's kind of gets inside of our middle ear and causes the motion of those bones to change in some way, that definitely affects our hearing quite a bit. But I haven't heard of degeneration of these joints. Maybe just because they're not like weight bearing, you know, there's not a lot of stress on them. They just kind of move back and forth on each other, but it doesn't mean that can't happen. I just haven't seen it. So yeah, it's a great question. Yeah. Other questions on that? Okay. All right, so this one just this is like a cross section of the middle ear, and you can kind of see a couple of those muscles right there and kind of where they attach. And yeah, and so we also have this little structure that connects the middle ear to the throat, right? It used to be called the eustachian tube. Now it's just called the name just includes the two kind of parts of the body that it connects, right? So the pharynx, which is your throat, and then the tympanic cavity, which is your middle ear, right? So it's called the pharyngotympanic tube. And yeah, it opens up when we swallow, yawn, or chew, right? So when your ears are kind of plugged and you kind of like move your jaw backwards, kind of open up your jaw, move that, you can feel that pressure change in your middle ear, right? And that's really what you're doing is you're kind of opening up that pharyngotympanic tube. And the reason for that is because the pressure inside your middle ear needs to be the same as the pressure on the other side of your tympanic membrane in the external ear, right? Because if it's not, so let's say let's say your pharyngotympanic tube is clogged up, right? Because you have an ear infection, and you've been down here at sea level, and then you decide to go take a drive up to Julian for the day, where it's 4,000 feet in elevation. The pressure is a lot lower up there than it is down here because you have less air between you and the top of the atmosphere, right? And so then the pressure in your external ear is much lower than it is in your middle ear, because your middle ear is still the pressure that was down here, right? And so since you got lower pressure outside, higher inside, it pushes your eardrum out, right? And then it makes it harder to move. It doesn't transmit, it doesn't move back and forth as well, so it doesn't transmit the sound as well. And so if your pharyngotympanic tube is open, you can just kind of move your jaw around, and then that equalizes the pressure, right? Because it lets the air in from the outside and makes it the same pressure that it was on the outside. But if it's not, if it's clogged up, then you're kind of stuck with that uncomfortable feeling of your eardrum being pushed one way or the other, right? But because of that pressure difference. Go ahead, Lisa. Yeah.
On the last slide, which was the bone that touched the eardrum?
Say that one more time.
Which was the bone that touched the eardrum?
Oh yeah, because that's that question there. Yeah. So we'll see it here in a sec, but we can see it right here too. So the one that looks like a little like stirrups is the one that goes all the way to the internal ear. So that's not the answer to that question. that's the one that touches the oval window, and then it's the one on the other side, the hammer shaped one right here that is touching the eardrum. Yeah. Good. All right. Other questions on that? Okay. All right. And then lastly, the internal ear. So once we get past that, Sometimes it doesn't move. There we go. Internal ear. Once we get past that last bone, we go through the what's called the oval window, and then after the oval window is where we start the internal ear. Okay, so these are tiny little delicate membranous structures, and it's completely encased in bone. Okay, so there's just little cavities inside of your cranial bones that house these little incredible organs that are so important for us. Okay, and so these are all fluid-filled structures inside. Okay, and there's 3 specialized regions inside the internal ear. Okay, we have the semicircular canals, which are these 3 loops. Okay, and these give us awareness of rotational movement of our heads. And there's 3 of them because in a way that really gives us 3 dimensions of rotational awareness. Right? So there's one kind of facing vertically, one kind of facing diagonally one way, one kind of facing diagonally the other way. So we can really pick up the rotation of our head in all 3 different dimensions. Okay, we have this little egg-shaped cavity that's the closest part to the oval window called the vestibule. And the vestibule gives us awareness of 2 different things, awareness of just the static position of our heads. Okay, so if we close our eyes, we still have a good idea of where our head's at. Right, in space. We can close our eyes and tip our head forward 90 degrees, and we pretty much are aware of ourselves doing that. We can sense that we're doing that, and a lot of that is because the vestibule is giving us information about that. And then the vestibule also gives us awareness of linear motions of the head. So just like straight back and forward, straight up and down, straight side to side. We'll talk about how those work more next time. And then the cochlea is this little snail-shaped canal that is where our hearing sensation comes from. Okay, and which cranial nerve innervates all these structures in here? There we go, vestibulocochlear nerve, right? And actually splits into the cochlear nerve and the vestibular nerve. And so the cochlear nerve goes to here, vestibular nerve goes to here. Right? Cranial nerve 8. Okay, questions about any of that? All right, I'll leave this up here just for a sec, but you can go ahead and start going to the next slide and start labeling all those structures we just talked about. And then, yeah, I know there's a clicker question on there. What does that say? Yeah, you're gonna send in a number to the answer to that question there. So the vestibulocochlear nerve goes to all of this, but it kind of splits into 2. There's the cochlear nerve that goes to this, and then the vestibular nerve that goes to both of those.
Okay, and then this one figures out like if you like that with your head.
Yeah,
but the other one is
spinning.
Just like I don't really get the difference.
Yeah, so linear motions are just like straight forward and backward, straight side to side. Rotational is like your whole body spinning.
Wait, it's your body or your head?
Your head is really where it's found. So
Okay, so head
yeah.
rotation and the other one's just.
Linear motions as well as static position. Yeah. Olivia, were you able to get the note packet yet? No?
It's ordered finally.
Okay,
Yes.
sounds good.
Yeah.
All right. Rex, are you still working on getting the note packet?
Yeah, I
Okay.
am. I sent some reach out, but I haven't got it.
Sounds good. Yeah, no problem. Just want to make sure this wasn't your long-term solution. But yeah. Okay, thanks.
No.
I had my clicker not signed up for the TBR.
Yeah, so
I did the steps that you sent on the announcement
when did you do it?
last week,
Okay, like right after I sent that out or?
the next day.
Okay, I haven't checked it for a couple days.
Okay.
But I'll check it again today and see, let you know if it's still not connected. So you put the clicker ID in and connected to the class. Okay, yeah, I'll check it out. I haven't checked it today, but I will, yeah. Annalise, are you still working on getting the note packet?
Yeah.
Okay. Sounds good. All right, guys, so you're sending in the number of the structure that gives awareness of rotational movements. Okay, just got to scroll through the numbers and then just go ahead and hit send. All right, give you about 30 more seconds to send that in. So you're doing a number and then hit the send button with this kind of question.
Five and eight.
Eight. Yeah, 5 is like the 6, 7, 8 are the bones. And then 5 is like the thing that bone is touching. All right, anybody else there on the clicker? I got 62. Might be most of you. Just make sure you hit that send button. Is that everybody? All right, so 89% of you are saying number 9 there. And all right, so we'll get down to that one in a sec. So let's come back together here. So obviously the 3 regions, right? We've got the external, middle, and internal. Okay. All right, number 1. Oh yeah, here we go. Number 1, we got the auricle, right? What's 2 called? Got the external auditory meatus. Okay. Number 3, tympanic membrane, right? Number 4, what do we call that? Pharyngotympanic tube. Okay. And then we get down to 5, which is just a little membranous structure that auditory ossicle is touching. It's the start of the internal ear. What's that one called? Yeah, so oval window. Okay. All right. And then we got the 3 bones. Okay. So starting with number 6 there, which is actually the one in the first one that's touching the eardrum. And what is that one shaped like? Kind of like a hammer, right? And you can kind of see how it's like, if this is the anvil, the hammer is kind of like hitting the top of it, right? Kind of, yeah. So 6 is the malleus, 7 is the incus, 8 is the stapes. Okay. And then we get to number 9, which is the one you guys chose for giving awareness of rotational movement. So that's the semicircular canals, right? And then we got this little kind of bulbous structure right here that's just on the other side of the oval window. That's called the vestibule. And then we've got the nerve that's already been split into 2 there. So what would 11 be? Which part of that nerve? What would we call it? Yeah, the vestibular nerve. Okay, and then we got the other part going to the cochlea, so that'd be the cochlear nerve. And then 13 finally is the cochlea. Okay. Any questions on any of those structures? All right, ready to move on? Everybody got it? And one thing I would highly encourage and ask you all to do is to just have your phones in your bags during class. It's just, I just know from my own self that if I have my phone out on my desk, even if I'm not using it, I'm thinking about it for some reason, you know. It's like our little friend that's there and wants to talk to us. But so if you just stick it in your bag, I think you'll be more engaged and you'll get more out of class time. Okay. All right, so let's talk a little bit more about some of the things going on here with these different parts of the ear. So I kind of already just mentioned it, but middle ear infections, also known as otitis media. Okay, so this is just something causes inflammation inside of our middle ear, right? And how many people grew up getting a ton of these as a kid? Yeah, we probably got some. Yeah, it's somewhat common, right? And then how many people actually had one so bad where it like burst your eardrum? Yeah, we got a few of those. How many people had the tubes put in their ears? Yeah, a couple of those too. Yeah, yeah. So this is somewhat common, right? Kids get a cold. That cold eventually, what happens is that cold's usually in your upper respiratory tract, right? So it's in your throat or it's in your nose. But then laying down a lot, some of that bacteria or virus can make its way through that pharyngotympanic tube up to your middle ear and kind of make a little home for itself up there, right? And that creates a bunch of pus and a bunch of fluid. And so what happens is this air-filled space turns into a fluid-filled space, right? And when those bones are trying to move through fluid, they don't move as well, right? They move a lot slower, so they don't, you can't transmit the sound as well. So it sounds like you can't hear as well. Everything sounds muffled, right? And especially when there's pressure in there, when you get enough pus and fluid in there, then it starts smashing the eardrum out and the oval window in, right? And then it gets really painful as well as really hard to hear. Right. So this is all kind of part of otitis media. Okay. One key kind of thing that can lead to this in babies is it used to be a thing to kind of like give your baby a bottle to help them fall asleep like while they're laying in the crib, but a lot of times the milk can actually just make its way up into the middle ear of the baby at that point. And it is more common in kids. One of the reasons is because there's just less distance from the throat up to the middle ear in kids than there is in adults. And so everything's just a lot closer. But yeah, some of those complications we talked about, ruptured eardrum. If this happens too much in children, then this can delay speech and language development. And if it gets really bad, this can get into your inner ear. If it gets into your inner ear, it can obviously lead to deafness, proprioception issues. But once you're in the inner ear, you're pretty much very close to the brain at that point, and it can actually make its way all the way into the brain. So if it's bacterial, then it'll be treated with antibiotics, and that can be taken care of pretty well for the most part. But yeah, and then if you just need to relieve pressure, this is what one of those tubes looks like. It's really just a little hole poked into the tympanic membrane to allow that fluid to release, so some of that pressure can drop. Okay, any questions about any of that? Okay, all right, so let's talk about a special structure that we find kind of commonly through all three of those structures within the inner ear. Okay, and it's called the hair cell. Okay, so the hair cell, again, is found in all parts of the inner ear. And it looks like this. So we have like a cell body with a nucleus, and then there are these little stereocilia on top, which are the little hairs. Okay. And these are going to be surrounded by supporting cells, just like we saw in the olfactory epithelium and the gustatory epithelium. And then the bottom is going to be connected to neurons of cranial nerve 8, right? So we can see those neurons connecting right there, supporting cells on the side, and the little hairs on top. Okay. And so depending on which one we're in, whether we're in the cochlea, the vestibule, or the semicircular canals, these hair cells are going to be embedded in something different, but they all kind of have in general, a similar function going on, to where something is moving those hair cells, and that's stimulating them to cause an action potential and send something down those nerves back to the brain. Okay, so those are really like mechanoreceptors is what they are, right? Because they're sensing mechanical pushing of themselves, and most time it's, or in the cochlea, it's vibration. In the other two, it's actually just fluid moving around. Yeah, and so that's kind of what those hair cells are. So we'll look at each of those structures that we just talked about, and just kind of talk about the different regions inside of those, and then in particular, where we find those hair cells inside of each of those. Okay, so we'll start with the cochlea. So the cochlea has really 3 separate canals inside of it. And we can see that here. There's the upper canal, okay, and that's the one that's going to connect to the oval window, right? So the upper canal receives sound from the oval window, okay? And then it's running parallel to the lower canal right below it, which is down here, okay? And that one's going to connect to a structure that we didn't mention on the other labeling side because we hadn't talked about it yet, but it's called the round window, which is the exit point of sound from the internal ear. So sound goes in the round window, or sorry, goes in the oval window, through this whole cochlea and then exits out the round window. Okay, so that lower canal allows sound to exit via the round window. Okay, so, and again, that's parallel to the upper canal. But then in between those two is this canal called the cochlear duct, right? And this is actually where we find the hair cells. Okay, so it's between the two, contains what are called the organs of Corti, or the spiral organs, okay? And in each of the structures that we look at, we're going to have a special structure inside of it that is the location of the hair cells. And so in the cochlea, it is the organ of Corti is where we find the hair cells. Okay, so the sound has to make us get into this middle canal for us to hear it. Okay, there'll be certain sounds that just go into the upper canal, go to the lower canal and exit out, and we don't hear it because they're not in the right frequency that our ears can hear. But if they're in the right frequency our ear can hear, then it'll go through that middle canal, stimulate those hair cells, and then into the lower canal and then exit out. All right, so let's look at that a little bit closer. So if we unroll the cochlea, which is what this image does, then it looks kind of like this. Okay, and so we can see those sound waves coming in. Those are going to shake the tympanic membrane. It's going to bounce back and forth. Right, those are going to move those bones back and forth. Since the bones are moving back and forth, it's going to move this oval window back and forth. And since there's fluid right here, by moving that oval window back and forth, that creates little vibration waves going through that fluid. All right, and so you can see those vibration waves are going to pass through. Okay, and the cool thing about this little membrane here on the bottom, which is going to be the bottom part of this cochlear duct, it's called the basilar membrane, and it's set up in such a way that the stiffness of it changes as you go from one end of the cochlea to the other. Okay, and so the stiffest part of it is the part at the very beginning close to the oval window, and then it gets kind of more flexible as we get down here towards the end. Okay, so what that means is the stiffer it is, that means it takes high frequency waves to vibrate it. Okay, but then on the tip of it, it's much looser and it only vibrates in response to low frequency waves. Okay, so when we get a high frequency sound wave coming in, what it's going to do is it's going to come in, it's going to vibrate the earlier part of the basilar membrane. Okay, and when it does that, then it crosses through the cochlear duct, so it's going to stimulate the hair cells in here. After it does that, it passes into the lower canal and then exits out. Okay, go ahead. I haven't heard that the basilar membrane changes in flexibility, but what is very common is for the early part, for you to have damage to your hair cells in the early part, because that just takes the brunt of a lot of those really loud sounds that we hear. And so, for example, when your ears are ringing, you hear a high frequency sound, right? You hear a high pitched sound. That's because you've damaged the early part of your hair cells, and those ones are telling your brain that you're hearing a high frequency sound. But I haven't heard the thing that it actually changes in flexibility. Did you see that somewhere? Okay, yeah, I don't think so, but definitely that's where we get damage most commonly, because that's just the first place that the sound comes in. So yeah, good question. Yeah, so then if we get a lower frequency sound wave, it vibrates much further down, right? So then it passes through the cochlear duct much further down, and then it can still exit. But then if we get a sound wave that isn't within that 20,000 hertz to 20 hertz range, then it's like we see here, then it's going to just go straight through. It's going to wrap around the corner here, make a U-turn, exit out, and we're never going to be aware of it. Right? So dogs, for example, can hear sounds of a much higher pitch than we can, right? So they have the first part of their basilar membrane is much stiffer than ours, and so they can hear those high pitched sounds that we can't, right? And so some of those, like those dog whistle sounds, just go right in and right out for us, but for dogs, they have a part of their ear that can hear that. So yeah, any questions about that? Yeah, Olivia. Go ahead. Yeah, we'll talk a little bit more about it next time too, but what it is typically those hair cells have been bent too far for some reason. Right? So you get some really loud sounds that come in that kind of smashes the hair cells to one side, and it can actually damage them in some way. And it can be temporary or permanent. But what happens is that makes those hair cells fire action potentials, or makes them fire, and then that causes the neurons connected to those hair cells to have action potentials. And that's what happens when the brain receives a signal from those hair cells on the real high pitched end, as we hear a high pitched sound. And so really those things are just always sending that sound into our brain. And so it's like we're always hearing a high pitched sound. And what really means we never get to hear that high pitched sound, right? So if that sound actually did come in, then we wouldn't actually be aware of it because we're already hearing it all the time. So yeah, that's pretty much what it is. Yeah. Other questions on that? Okay, so just a little bit more about this organ of Corti. So we kind of already talked about the basilar membrane and what it does. So this is the floor of the cochlear duct. Okay, this is the bottom part of it. This is where the hair cell is anchored to. Okay, this is the one that's actually going to move with the sound waves. Okay, and again, the side closest to the oval window is the stiffer part. The side at the apical end, or the furthest away from the oval window, is more flexible. Okay, and the function of this one is to support that spiral organ and anchor those hair cells, but also allow for that movement. So it can move when sound waves vibrate through it. Okay, so that's the bottom, but then the top where the hairs are sticking up, we have another membrane called the tectorial membrane. Okay, and this is like the ceiling of the organ of Corti, and this is where the hair cells are embedded into. Okay, so this one doesn't move at all. And so the function of this is for like a stiff surface for the cilia to move against. Okay, and so if you picture the organ of Corti like this, like the bottom of my hand is the basilar membrane. Okay, and this top is the tectorial membrane. We have the little hair cells sticking up like this, right? And so the tectorial membrane holds those tightly. And when sound moves the basilar membrane, this one stays put, but the other one moves. So that bends the hair cells back and forth. And so that's really what we need, is kind of, if they both move together, then the hair cells wouldn't actually move, right? So we have one stiff one on top, and then the one that moves on the bottom, so the hair cells actually move. Yeah, go ahead. Yeah, so the bottom of them are anchored in the basilar membrane, and then the hairs themselves are anchored in that tectorial membrane. Yeah. Good. Other questions on that? Go ahead. All right. So the basilar membrane is the one that moves. So this is the one that allows some movement, whereas this one, the tectorial membrane, provides a stiff surface to the cilia for the cilia to move against. Good. Other questions? Okay. All right, so let's take a second and just have you guys label these, and then you can answer that question there on your clickers.
C would just be the hair. Yeah. Oh, wait.
All right, so you're sending in the answer to that question there. Which is the one that moves? All right, about 15 seconds. Go ahead and send it in if you haven't yet. All right, anybody else? Is that everybody? Good, looks like about it. All right, so about 80% of you are saying B is the one that moves. So let's walk through those here. So A is pointing to that membrane on the top there. What do we call that one? Yeah, that's our tectorial membrane. And then B down there is one you guys said is the one that moves. So that's our basilar membrane. That's correct. And then C is just pointing to the hairs themselves, right? And then D is just pointing to those hair cells. And there are there's actually inner hair cells and outer hair cells. We don't need to get into the difference between those in this class. And there is actually efferent fibers going to the cochlea as well. So that's why it specifies afferent. But again, we won't get into those details of what those do for the purposes of this class. So, any questions about those structures? Give you a sec to finish writing those down. And one thing I highly encourage is for you to make a decision when you're filling these out. And so that what I'm saying by that is instead of waiting for me to uncover them, you write down your best answer. Okay, because there's actually a lot of research in education showing that just making a decision, whether it's right or wrong, while you're in the learning process, helps you learn much more than it does to just wait until you see the answer and then write it down. Okay, and that's why I use the clickers. That's why I do a lot of fill-in things like this. So just kind of some of my philosophy behind that. Okay. All right, so let's look at the next structure, the vestibule. Okay, and the vestibule again is helping us with those linear movements of the head, as well as the position of the head with respect to gravity. Okay, we have kind of three different names here to wrap our heads around. We have these two little structures that are the same, just in different positions, called the utricle and the saccule. Okay, these both inside of them contain what's called a macula. Okay, and the macula is the place where the hair cells are in these in this vestibule. Okay, and then inside of those, or sorry, then inside the vestibule we have the utricle and the saccule, and then inside the utricle and the saccule, each there's a macula. Okay, lots of strange words for sure. But what we find in the macula is these little structures called the otolithic membranes, as well as the otoliths. Okay, so oto means ear. Lith or lithic is referring to a rock of some sort, right? And we'll see on the next slide that it's literally what it is these little stones embedded in a membrane that give some weight to that membrane to allow it to move when our head moves or when our head changes position. Okay, and so the difference between the utricle and the saccule is that the utricle, when we're standing upright, is parallel to the base of our skull, but then the saccule is perpendicular. Okay. And we really need both of those in order to maintain awareness of our position and our linear movements. Okay, and so the way that we bend the cilia with this structure is again, let's say, so if we're talking about the utricle, the one that's parallel to the base of the skull, it's sitting up like this. It's got the little rocks on top embedded in the membrane, and you can see those here. Okay, you can imagine when you start moving forward, there's gravity that kind of pulls those rocks backwards, right? And that's really what happens is the little rocks give that membrane enough weight to go backwards when you go forwards, and that bends the cilia, right? Same thing if you take your head and you just tip it forwards, right? Those little rocks give that membrane enough weight to pull down, right? And that stimulation tells your brain that your head's moving forward, okay? Yeah, so that's pretty much the basics of that. Okay, so take a sec and label these structures, and then you can send that the answer there in.
I was just thinking because it's like a. I was just thinking.
So I think to differentiate that one is more like. No, I think bending. Oh wait.
All right, so you're sending in a letter there. You will have to hit send. All right, anybody else? Give you about 15 seconds, send that in. Make sure to hit the send button. Since F was up there, I made it that type of question. There we go. Okay, good. I think we got everybody. So we got about 46% of you saying B. I think that is our biggest one. And then about 17% saying A. So yeah, I kind of walked you through the one with a forward movement, right? So if we have two of the structures like this, the one, and we got the rocks on top of each, right? The one with the forward movement, that's going to move backwards when I move forwards, right? But when I go straight up, that one doesn't really do anything, right? Because it's got rocks on top, just go straight up and down. But the one that's like this, got those rocks pulling it down. When I jump up, the rocks are going to kind of go the opposite way, right? It's going to pull it down. Right, so we're thinking about the one that's perpendicular to the base of the skull when we're talking about an up and down motion like that. Right, and what's that one called? The one that's perpendicular? The saccule, yeah. So A is the utricle, B is the saccule. So B would be the correct answer there. Okay, questions on that? And we will dive into that more next class too. Yeah. Okay, so then C is just those stereocilia again. D is the little rocks themselves, the otoliths. And then E is this like jelly-like membrane that holds those rocks. And there's also what the hair cells are embedded in, so the otolith membrane. And then F would be neuron fibers from which nerve? That same one, right? Cranial nerve 8, vestibulocochlear nerve. Right? And specifically within that, the vestibular nerve. All right, questions on any of those structures? Okay, all right, so then let's look at the last one. And the last one is the semicircular canals. And again, these are helping us with being aware of rotational movements of our head, okay? And specifically, it's change of rotational movements. We'll talk more about that next class. But again, there's 3 canals, they're fluid filled. The special structure in here that we'll look at is called the cupula. Okay, and the cupula is this tall gelatinous structure that kind of sticks up at the base of the loop, right? So it's kind of sticking up at the base, and there's the loop canal around it. Okay, and the cilia are embedded in the bottom of that cupula. Okay, and so the way that the cilia are bent in the semicircular canals is that fluid is literally just moving rapidly through the canal. And when it pushes up against that cupula, it kind of bends it one direction or the other, depending on which way the fluid is moving through the canal. Okay, so we can see these structures here. A is our cupula sticking up. We can see those hair cells embedded right in the bottom of it. And then we can see those stereocilia sticking right up into the cupula. And you can see how fluid would be wrapping around here fastly. When it does that, it moves the cupula one direction or the other, and that's what moves the hair cells. We'll get into the details of how that works and how our body interprets that on Wednesday's class. Okay. All right, so last slide is just a recap. So I want you to work with your team and just kind of fill in that table based on those 3 structures we just talked about. And then we'll come back together for a sec and wrap it up.
Sound is like received in the middle here, right? Like that's where
The
it's
middle
like
canal, that's when we actually are aware of it.
Oh, yeah.
So that it first goes through the upper canal,
And then that's where
and then if it can bend the basilar membrane, then it'll pass through that middle canal.
So like, does it send like back to the lower canal or just goes to there?
Yeah, it does. It's better on this one because it goes in, and then if it can move the basal membrane, it passes through the middle part,
Okay.
and then back out
What
that
if
way.
it's like below like 20 hertz? Like, would you even like pick it up in here? Like, does it have to go in there?
It goes in there just because it vibrates your eardrum. Yeah,
Okay.
and so it just goes around and comes back up.
Okay.
Yeah, but it does, since it's sound, it's still going to have some energy to be displaced on your eardrum. So yeah.
Or the bending of the cilia.
Yeah.
All right, guys, since we're just about out of time, let's go ahead and just finish this up here. So one key part of each of these is what the little region is called, where we find the hair cell. So what is it in the cochlea? Organ of, the organ of Corti. Okay, that's where the hair cells are. In the vestibule, what do we call those ones? There's the utricle and the saccule, but those both contain a what? Macula, okay? So that's where the hair cells are there. And then in the semicircular canals, the hair cells are embedded in the cupula. Okay, so the function and stimulus that they're sensitive to are kind of similar. So obviously this would be sound. This would be linear movements, or really just position. All right, and semicircular canal is rotation. Okay, so then obviously the function is hearing. And then these ones are just awareness of those, right? Awareness of those two things. All right, guys, I'll leave this up here for a sec. But have a great rest of your day. We'll see you guys on Wednesday.